Hi guys, welcome to my video. In this video is a continuation of the part 1 video about the protein synthesis, the translation. Right now, we will discuss the second process, which is the translation. So, discovering this sequence of events was a major milestone in molecular biology. It is called the central dogma of biology. The two processes involved in the central dogma are the transcription and translation. So let's move on to the translation process. The translation is the second part of the central dogma of molecular biology, from RNA to protein. It is the process in which the genetic code in mRNA is read to make a protein. Remember that mRNA comes from the transcription in the nucleus. The translation is illustrated in the diagrams below. After mRNA leaves the nucleus, it moves to ribosomes. It moves to ribosomes, which consist of rRNA and proteins. Translation happens on ribosomes floating in the cytosol, or it could be the ribosomes attached to the rough endoplasmic reticulum. The ribosomes read the sequence of codons in mRNA, and molecules of transfer RNA bring amino acids to the ribosomes in a correct perfect sequence. Just as with mRNA synthesis, protein synthesis can be divided into three phases: initiation, elongation, and termination. In addition to the mRNA template and ribosomes, many other molecules contribute to the process of translation, such as tRNAs and various enzymatic factors. To understand the role of transfer RNA, you need to know more about its structure. Each transfer RNA molecule has an anticodon for the amino acid it carries. Note that the anticodon is a complementary to the codon for an amino acid. For example, the amino acid lysine has the codon AAG. So the anticodon is UUC. Therefore, lysine would be carried by a tRNA molecule with the anticodon UUC, which is they are perfectly paired. Wherever the codon AAG appears in mRNA, a UUC anticodon of tRNA temporarily binds. While bound to mRNA, tRNA gives up its amino acid with the help of rRNA. Bonds form between the amino acids as they are brought one by one to the ribosomes, creating a polypeptide chain. The chain of amino acids keeps growing until a stop codon is reached, so there is a stop signal codon that can be reached. Ribosomes, which are just made up of rRNA and protein, have been classified as ribozymes because the rRNA has enzymatic activity. The rRNA is important for the peptide transferase activity that bonds to amino acids. Ribosomes have two subunits of rRNA and protein. The large subunit has three active sites called E, P, and A sites. These sites are important in the catalytic activity of ribosomes to make proteins. Just as with mRNA synthesis, protein synthesis can be divided into three phases: the initiation, elongation, and termination. In addition to the mRNA's template, many other molecules contribute to the process of translation, such as ribosomes, tRNAs, and various enzymatic factors. Now, let's move on to translation initiation or initiation of translation. In this process, the small subunit binds to a site upstream on the 5' side of the start of the mRNA. It proceeds to scan the mRNA in the 5' to 3' direction until it encounters the start codon, which is the AUG. Remember that AUG is the start codon, and it makes a start of the synthesis. The large subunit attached in the initiator tRNA which carries methionine, binds to the P site on the ribosomes. In elongation, the ribosomes shifts one codon at a time, catalyzing each process that occurs in the 3 sites. With each step, a large tRNAs enter the complex, and then the polypeptide becomes one amino acid longer, and an uncharged tRNA departs. The energy for each band between amino acid is derived from GTP. A molecule similar to ATP, the GTP. Briefly, the ribosomes interact with other RNA molecules to make chains of amino acids called polypeptide chains due to the peptide bonds that forms between individual amino acids. Inside the ribosomes, 3 sites participate in the translation process, the A, P, and E sites. Amazingly, the E. coli translation apparatus takes only a massive 0.05 seconds to add rich amino acids, meaning that a 200 amino acid polypeptide could be translated in just 10 seconds. That's way awesome. In termination, translation in termination of translation occurs when a stop codon, the stop codon are UAA, UAG, or UGA is encountered. When the ribosomes encounters the stop codon, the growing polypeptide is released with the help of various releasing factors, and the ribosome subunits dissociate and leave the mRNA. After many ribosomes have completed translation, the mRNA is degraded so the nucleotides can be reused in another transcription reaction. Remember that in the initiation phase, the initiator, tRNA, carrying methionine with its anticodon encounters the AUG start codon at the P site of the ribosomes. During elongation, the ribosomes translocate in the 5' to 3' direction, at which point the amino acids attached by peptide bands to the tRNA in the site and can be bound to the tRNA in the A site, creating a long amino acid chain based on the codons in the RNA. As the ribosomes translocates, the tRNA leaves the ribosomes through the E site while new tRNA enters the A site in order to continue elongating the amino acid chain. Then, after a polypeptide chain is synthesized, it may undergo additional processes. For example, it may assume a folded tertiary shape due to interaction among its amino acids, and it may also bind with another polypeptides or with different types of molecules, such as lipids or carbohydrates. It changes its forms at times, and many proteins travel to the Golgi apparatus within the cytoplasms to be modified for the specific job they will do. So that's it for protein synthesis. I hope you've learned something. Stay safe. God bless.